Dr. Kurgonda for the introduction, and good morning, everyone. I'm very happy to see you again today, and as you can see from the program, we again have a very wide range. We are going to cover a very wide range of topics today, and sometimes that means we perhaps will be listening to something that is not exactly in our area. But sometimes a topic from other area can bring inspiration to us. So I'm really interested and exciting to listening to every single talk today to make the best of it. And again, because conferences is also about introduction and making connection and potentially establish collaboration. So I would like to first spend a few minutes to introduce the research program in my laboratory. I have two research programs in my lab. One of them is more focused on reproduction and oocyte competency, which is not very relevant to this conference. The one that is more relevant to this conference is concerning the early wean animal and colitis animal. Let me see whether I can make this work. There you go. In the pig industry, pig now these days have been weaning earlier and earlier. They are typically weaned between 7 to 8 weeks younger than they naturally supposed to. This is done because we need to allow the mother to return to the production line, as well as to prevent the spread of disease from the mom to the baby. However, this early weaning, of course, would cause the intestine stress and therefore associate growth rate drop. And because the premature weaning, the intestine is not well developed. That's why they are more susceptible for infection, which is a very similar situation like colitis in human. And in the person, antibiotic being included into the feed of the animal to serve as therapeutic purpose and growth stimulator. However, perhaps a lot of you have aware that the use of antibiotic in the feed has been banned. And it will be banned in the North America, maybe other countries as well, sooner or later. That's why alternative help is required. And in my program, I'm looking at this problem from another window. Instead of just trying to kill the enemy, which is the pathogen, we are hoping to build a stronger intestine and a stronger immune system to defend, to allow this pellet to defend. So upon intensive literature search, we have found that, and also because the root of the problem is because of the sudden stop of milk supply. So we identify a few factors that are rich in the milk and are important for intestine development, which are this growth stimulator, as well as some of the tissue repair factor. And that is when the intestine has been stressed, and in fact, they help the repair of the tissue there. And these factors have been, in the past 20 years, demonstrated to be very important for animal growth and intestine and human as well development. However, the currently markedly available factor is too costly to be applicable for animal production or even for human application. So our first goal was to establish a system to produce this factor in a much cost-effective manner. And in my laboratory, we have established two expression systems using antimicrobial. The first one is called Lactococcus lactis. It's a food-grade bacterial that we use in the fermentation industry a lot, such as yogurt, pickle, and so on. And in the pig, this is also a probiotic. So we are hoping one stone killing two birds using this system to deliver the bioactive factor we want to deliver, at the meantime providing the beneficial material to the animal. And another system we use is the yeast system, which have a better expression capacity, and the medium is cheaper. So using these two systems, we have been able to generate, using EGF, which is in full name epidermal growth factor, as an example. As you can see, we generate the pig EGF in L-lactis, Lactococcus lactis, as well as yeast in a secrete form, so that even if we just feed the suppurator to the animal, that already contains our bioactive peptide. So after the production, we try to test whether the EGF we produce with the bioorganism is active using a cell proliferation assay. As you can see, supplementation of the EGF we produce was able to successfully stimulate the proliferation of the cell, which is the normal physiological function of this growth factor. And also, in addition, we also study whether these yeast-produced factor are functional in the animal, in vivo, by using animal study. In this particular study, we have two large groups. The first two, which is in white and blue, the animal feed to the early wean pig with the very cheap and low protein feed. And the green and red are the ones feeded to, they are feeded to the more expensive feed. We use BP, stand for black plasma, which is a very high quality and expensive protein. And as we can, and then they are, in each of the group, they are feed in the absence and presence of the EGF we generate. As you can see, starting from week 2, feeding of EGF start to increase the body weight gain of the pig, and this enhancement was more significant when it reached the week 3 after weaning. So suggesting the growth factor we produce is functional. And this slide, we are just hoping to look at the mechanism why feeding our factor allow the pig to grow better. So villi high is one. Villi is one of the tissue structure in the intestine that is important for digestion and absorption. So when we measure villi high, we see that EGF enhance the development of villi, making it longer. And also, when we look at the inflammatory factor, laminar thickness, as well as some of the anti-inflammatory cytokine, we see the decrease of this inflammation factor, the inflammation factor and the anti-inflammatory cytokine expression. So telling that EGF work well to the animal because it stimulates the intestine development, at the meantime suppress inflammation there. So those are just one of the examples of the potential application. And because of the tissue stimulating nature, EGF can be also used in wound healing, dressing, supplement for animal or human colitis. And in some of the company, the cosmetic company, this is one of the important factors included in the face cream, because then you can stimulate the renewal of the skin and making less wrinkle. And okay, now I'm switching to the major topic I'm going to talk about. So I would be talking about production of recombinant antimicrobial peptide using food-grade microorganism. Why antimicrobial peptide? The therapy of infectious disease is currently under severe threat because of two reasons. Number one, we have been using a lot of antibody, both in animal production and in the hospital. So generating more and more antibiotic-resistant material. However, at the meantime, very few novel antimicrobial compound has been generated and entered the clinical use. Therefore, we have an urgent need for anti-infective alternative. So that's why we are looking into other small peptide. And antimicrobial peptide, they are very small, making it a good candidate for therapeutic purpose, and produced by animal cell as opposed to the real antibiotic, which is produced by bacteria. They have broad spectrum of antimicrobial activity. That means they can kill both gram-positive and negative bacteria, and they are actively even against the bacteria that has antibiotic resistance. So the reason that they are working so well is because of the major mechanism of the antimicrobial peptide is different from the currently used antibiotic. They kill bacteria by interacting with the bacterial membrane through the negative-positive charge interaction, and they are causing the rupture of the bacteria. Therefore, in order for the bacteria to generate resistance to the antimicrobial peptide, they have to change the membrane, which require the mutation of many genes. As opposed to antibiotic, they require the mutation usually of one single gene. And also, more excitingly, in addition to just killing bacteria, they also have immunomodulatory effect. That means they can stimulate the immune response and motivate cell migration and help the tissue repair when they are killing at the same time when they're killing the enemy, which is the bacteria. So the antimicrobial peptide we have been working on is called Protectin-1. It's a pig-originate antimicrobial peptide, one of the most potent antimicrobial peptide identified so far. And that's why we are excited, because even in the human pharmaceutical company, they are trying to synthesize this one as well because of the potent activity. And they're known to have activity against bacteria and even some of the viruses, such as HIV virus. And we are hypothesizing that in addition to this, they also have immunomodulatory activity, and that's something we would like to test with our system. So for the Protectin-1 gene, it's a component of the innate immunity. It belongs to the cathelicidin family, and the same family member in the human and mouse form have been known to have immunomodulatory effect, although the effect for the pig form is unknown. And this is the gene structure of Protectin-1. It composed of the cathepsin and mature PG1 domain and linked by an enzyme cleavage site. And cleavaging of this proform would generate a cathepsin domain and mature PG1. And now it's known that the mature PG1 is the one having antimicrobial activity. However, the function of these two are unknown, and that's what we are hoping to find out using our system. So in order to study the function and application of this antimicrobial peptide, we automate the codon. We automate the codon of pig EGF to make it suitable for yeast expression and translation, and introduce this new DNA into yeast, generate different kind of yeast, and through this automate fermentation procedure, we were able to generate and secrete pig protectin 1. So this is ELISA and Western blot showing that the just using proform protectin 1 was generated, and after cleavage we also result in getting the mature PG1 as well. This is a negative and positive control. To study if the PG1 we generate is functional, we perform a radiodiffusion assay, which is an assay to test the antimicrobial activity. If they have antimicrobial activity, it will generate a clear zone in the middle. That means they're killing the bacteria around. So as you can see, after cleavage to generate the mature PG1, we were able to demonstrate antimicrobial activity. And interestingly, the MIC, which stands for minimum inhibitory concentration, are the same from what we generate by yeast and the one that is synthesized and commercially available. And this, we also study the stability of the product, because this is very important for application. So as you can see, by the same assay, antimicrobial assay, we were able to see that the mature PG1 was active. You see the clear zone here, active across a wide range of pH, and crossing a wide range of temperature as well, from 25 to 95 degrees, suggesting that it would be a good application potential of this small peptide. So to answer our research question about whether this antimicrobial peptide has other function, one of the one we asked is whether this PG1 can stimulate cell migration. We use a cell migration assay, a very simple and interesting assay, in which we culture the cell in a chamber like this, and this has a membrane. So if the reagent has a ability to stimulate cell migration, they will be able to migrate from one side of the membrane to the other side. So after 24 hours of treatment, we collect the cell from the other side and use horse staining, and then we were able to see whether cell migration was stimulated by the treatment. And as each of the blue dots represent one cell. As you can see, compared to the untreated control, each of the PG1 component has ability to stimulate the migration of the cell. So then we test the PG1 activity in vivo by using a dextran sulfate sodium-induced colitis model, which is a chemical that in human and mice very commonly used to induce colitis for inflammatory bowel disease. And so with this model established, we test different components of PG1 as well as control, and looking at a few index of colitis. The first one is the body weight loss, because you perhaps can imagine when people have diarrhea and colitis, they affect the absorption function, right, and digestion function, so they have body weight loss. And as you can see here, without any treatment, just by inducing colitis, the green line indicates the body weight gain, the body weight of the animal drop, like decrease gradually. However, this decrease was prevented by treatment of the antimicrobial peptide. Compared to this healthy control without colitis, it is still not the same, but they are quite efficiently prevented. And also, we measure the effect through the measurement of disease activity index, which is the combination of stool consistency, stool blood, and body weight. And this is a very standard index for measuring whether the colitis patients are responding to treatment effectively. And the higher the index means the more severe the symptom. So as you can see, without any treatment in colitis, these two are the negative control. The disease index will increase significantly. However, this increase was prevented by the treatment of the three components of the protectin 1, indicating that protectin 1 can suppress the symptom of colitis, help to improve the treatment. So to further examine, investigate why putting this treatment allow us to have such a good therapeutic effect, we look a little bit back into the intestine of the animal, and as you can see, by measuring this is the large intestine ring. Even if you just look at by visually, you will be able to see that this is the healthy control. This is the healthy control. This is the colitis without treatment, and this is the colitis with treatment. You can see that colitis with treatment of the antimicrobial peptide is looking very similar to the control. Retain the intestine ring. Similar effect was also observed when we are measuring the weight of the intestine. And I don't expect you to know how to read the histopathology, but just to put it in a very simple way. This is the histo section of intestine. When it's normal and healthy, this is how they look of the intestine. And when there is colitis, this is how they look. As you can see, the architecture of the V-line has been destroyed, and there's inflammatory index here. However, if you induce colitis and treat it with different form of protectin 1, you will see that they were able to help to restore the normal structure of the intestine to very similar to the one that is healthy control, right, compared to this one. And these two slides are just looking, we are just trying to investigate the molecular mechanism in terms of molecularly, what is the mechanism in giving us this very beneficial response effect. And by just this is one of mucin 2 is a protein that is providing a protective layer on the intestine. And as you can see, this is the healthy control. When there is an induction of colitis as a self-protective effect, the colitis induced the production of this protein. And this protective protein was further enhanced by PG1 and pro-PG1, although no effect in the catalytic domain. Similar effect was also observed in the tissue factor, tissue repair factor expression. And this one, perhaps I'm not going to go into great detail because the audience are not in this particular field, but we are also measuring the inflammatory response, like suppression of inflammatory response. They both are inflammatory factor. As you can see, compared to the negative control, we have a suppression to a different extent of the inflammatory factor expression by the feeding of the intestine to the animal. So in summary, the protectin 1 we produced by the yeast was able to protect mice against the DSS-induced colitis. And the histo and gene expression results are reflective of the phenotype observed. And these are just some molecular mechanism. Therefore, our conclusion is recombinant PG1 has immunoprotective effect in colitis, in DSS-induced colitis mice. Again, what I have presented is just one of the potential application of the antimicrobial peptide we produce. We can, many other potential application that we are going to look for industry partner in the future. This could be a very good alternative for antibiotic in terms of application, especially to the antibiotic resistant infection, because they kill also antibiotic resistant gene bacteria, and it could be used as wound infection dressing, external use should be a very good, effective one. And colitis therapeutic, and also can be used for food preservation to prevent the food from being infected by, being spoiled by the bacteria. And with that, I would like to thank all my collaborators and my student and the funding agent for my research. Thank you.
Thank you very much for your presentation. Now the presentation discussion for open discussion. If there are any questions.
Did you check it for the antifungal activity also, as it's an antimicrobial peptide? So did you check it against some fungi also?
Fungi, yeah, that's a good one. We don't have in theory they should have antifungi activity as well, but then I don't have a fungi assay in my lab, so I didn't check that.
But you have said that it's an antimicrobial peptide, so I thought maybe you have checked it for fungi also.
Yeah, people have already demonstrated that in the native form.
Okay.
But hopefully this one also have the same activity as well.
You intend to use it as say one of the potential uses is as a food preservative.
Yes, food preservative, that's right.
Don't you think it might have some negative effect on the food?
This antimicrobial peptide is pretty specific to bacteria because of the charge. It was very positively charged, so they are more they have more affinity to the pathogen. But when if it's used in a very high concentration, there is an opportunity that they can also bind to mammalian cell. But that the mammalian cell have a protected layer of cholesterol on top. That's why the chance would be low. But if you, for instance, we are thinking about ham and all, putting it in the ham for preserving the ham and things like that. For that, in that case, when by the time it was eaten, it was fully diluted. That they perhaps become the physiological concentration of part of the animal or human. So we are thinking, hopefully it will be safe, but it's an important question to ask. We need to test it before giving to the industry.
What is the size of the small peptide size?
The size? For the small protectin one, it's about 21 amino acid. The protectin one is 121, I think.
Do you characterize what are the amino acids present in that? Amino acids. Which are the amino acids involved in the polypeptide chain?
The amino acid?
Amino acids.
Yeah, that's the amino acids number. Like 121 is the full length of protectin one.
Yeah.
Did you test the mechanism of action which this peptide follows, antimicrobial peptide? The mechanism of action which it follows?
Yeah, the mechanism of action for this is a very strong positively charged peptide, and the bacterial surface is negatively charged. So they interact, and through the for this particular one is through they form a lot of pore hole and cause the osmotic change and allow killing the bacteria.
So if there are no questions, on behalf of Omics International, I feel it is a pleasure to honor her by giving this momentum.
Oh, thank you so much.